This is Chapter 3, Module 3, Cellular Transport. The learning objective of this module is to describe the different modes of transport of substances across the cell membranes. The phospholipid bilayer, the proteins, and the carbohydrates all play an important role in providing a barrier for the cell from its external environment, but it also helps to bring nutrients into the cell and to get the waste products and cell secretions out of the cell. How easily things can get in and out of the cell is called permeability. If a membrane will not let anything in or out of the cell, that membrane is considered impermeable. If a membrane allows anything to pass through it at any time, the membrane is called freely permeable. A selectively permeable membrane is a membrane that will restrict movement in and out of the cell, but movement is still allowed. The plasma membrane is a selectively permeable membrane. The plasma membrane will allow some materials to move freely, but restricts other materials from moving in and out of the cell. The plasma membrane determines which materials will cross, how they will cross, and when they will cross the membrane based on several factors, including the size of the material that wants to cross, the electrical charge of the material, the shape of the material, and whether the material is lipid-soluble or not. If the material is lipid-soluble, it will be able to cross through the phospholipid bilayer. But if it is not lipid-soluble, it will have to pass through the integral protein channels. The transport of materials across the plasma membrane will either happen without energy being used or it will require energy. If the transport requires energy or ATP, then the transport is considered active. If no energy or ATP is needed, the transport is considered passive. Diffusion is an example of passive transport. Vesicular transport is an example of active transport. And carrier-mediated transport using a carrier protein can be either passive or active. Diffusion is based on the theory that all molecules are constantly in motion. In solution, molecules will move around randomly, bumping into other molecules and randomly mixing in with all other molecules. These molecules are considered the solute in the solution. When more of one solute is found in one part of the solvent than in the other, this is termed a concentration gradient. Due to the random collision of molecules, the molecules will move from the area where it is more concentrated to the area where it is less concentrated. This movement of a solute from an area of higher concentration to an area of lower concentration is called diffusion. It happens passively and no energy or ATP is needed. An example of everyday diffusion outside the body is when perfume or Febreze is sprayed in a room. The scented molecules will start to bump into each other and begin to spread around the room. The students in the front of the class will be able to smell the molecules first, and after a minute or so, the students in the back of the class will be able to smell the molecules as well. This is due to the random passive collision of molecules in the air. In the body, an example of diffusion is when small nonpolar molecules cross the phospholipid bilayer. These molecules cross the plasma membrane down its concentration gradient. A concentration gradient is the difference between the high and low concentrations. Moving down a concentration gradient means the molecule will move from the area where there is a higher concentration to an area of where there is a lower concentration of these molecules. They will continue to move until the concentration on both sides of the plasma membrane are equal. Several factors influence how fast diffusion will take place across the plasma membrane. The distance the particle has to move is one factor. The further the particle is from the membrane, the longer it will take to diffuse. The size of the molecule matters too. Smaller molecules can diffuse more rapidly than larger molecules. Temperature is also a factor. Molecules move faster in warm temperatures. And electrical forces are a factor as well. Opposite charges attract, so a positively charged molecule will be attracted to the negative charge inside the cell and will be able to diffuse more quickly than a negatively charged molecule which would repel from the negatively charged molecule inside the cell. Across the plasma membrane, diffusion can be either simple or channel-mediated. Simple diffusion is when materials diffuse through the phospholipids without requiring specific channels. Lipid-soluble compounds such as alcohols, fatty acids, and steroids are all able to simply diffuse across the phospholipid bilayer and into the cell. Dissolved gases such as oxygen and carbon dioxide can also diffuse across the phospholipid bilayer through simple diffusion. Water-soluble compounds and ions are unable to cross the phospholipid bilayer. They will have to cross the membrane through protein channels. The size of the compound and the charge of the ion will determine when and how fast these molecules will diffuse. Channels that allow the free movement of these ions are called leak channels. Leak channels are always open and allow a slow movement of ions through them, allowing these substances to enter and exit the cell at a slow but steady rate. For example, we learned that the concentration of sodium is higher in the extracellular fluid than it is in the intracellular fluid. From our discussion on diffusion, we realize that sodium will try to move from the outside of the cell where its concentration is higher to the inside of the cell where the concentration is lower. Sodium will move down its concentration gradient and into the cell through these leak channels. Likewise, potassium will move through its leak channels out of the cell where there is a lower potassium concentration. Osmosis is a special kind of diffusion where water diffuses across the plasma membrane. Water will move to areas where there is a higher concentration of solutes in order to balance the concentration of solutes on either side of the membrane. To explain this, a solution that has more solute molecules has a lower concentration of water, and a solution that has fewer solute molecules has a higher concentration of water molecules. If the membrane is freely permeable to water but selectively permeable to solutes, then water molecules can diffuse across the membrane toward the solution with more solutes. When the water molecules move toward the side with more solutes, the volume on that side of the membrane will increase. However, the concentration of solutes in solution will even out. Osmotic pressure is a term given to the force of this concentration gradient of water. Think of osmosis as water always wanting to follow the higher solute concentration, and think of osmotic pressure as how much force that pull towards the solutes is. An opposing force to osmotic pressure is hydrostatic pressure. Hydrostatic pressure will push water molecules against the osmotic pressure. We will discuss osmotic pressure and hydrostatic pressure in other chapters. Let's consider cells in solution. The fluid inside the cell, or intracellular fluid, has a certain number of solutes within it. The fluid outside the cell, or extracellular fluid, has a certain number of solutes within it as well. If the concentration of solutes outside the cell is equal to the concentration of solutes inside the cell, then the extracellular fluid is considered isotonic. When a cell is in an isotonic solution, water molecules will freely cross both ways, in and out of the cell, but the concentration of the solution on either side of the membrane stays the same. If the cell were placed in a hypotonic solution that has less solutes than the number of solutes inside the cell, the water molecules will move inside the cell toward the solutes. When water moves inside a cell, the cell begins to swell and may eventually burst from the water pressure inside. The cells in your skin may swell when you take a bath in pure water. Pure water has a low solute concentration and therefore is a hypotonic solution. The solution outside of the cell loses water through osmosis as the water moves into the cell. In contrast, if the same cell was placed in a hypertonic solution, where there were more solutes in this solution than there are solutes inside the cell, then water from the cell will move toward the solutes in the solution, and water will leave the cell. When water leaves the cell, this causes the cell to shrink or crenate. Therefore, crenation occurs when the cell is placed in a hypertonic solution. The cells in your skin may shrink when you swim in the salty ocean, which has a high solute concentration, and therefore is a hypertonic solution.
What happens to a cell when it's placed in a hypotonic solution versus a hypertonic solution? A hypertonic solution is simply a solution that has a lower concentration of solutes, dissolved substances, compared to the inside environment of a cell. For example, water is considered a hypotonic solution since it is very low in solutes. A hypertonic solution is a solution that has a higher concentration of solutes compared to the cell's inside environment. For example, water with a lot of salt is considered a hypertonic solution since it is very high in solutes. How can we examine the effects of a hypertonic solution on a cell? You might be thinking, well, I'm made out of 50 to 70 trillion cells. If I drink this, I'll know what happens to cells in a hypertonic solution. But do not do that. Drinking salt water can be very dangerous as it severely dehydrates you. Nausea, vomiting, and even hallucinations are just a few symptoms of drinking massive amounts of salt water. Ask anyone who's been stranded at sea, which is unlikely because people who drink seawater eventually died due to kidney failure. So what you really need is a lab animal to conduct your test. Darn, she's too smart. Time to try her older brother. Well, that was a bad experiment. What's a better way to test how a cell reacts when placed in a hypertonic solution? Some eggs, like chicken eggs, are surrounded by a hard outer membrane which protects the semi-permeable inner membrane. This membrane is semi-permeable because it allows substances to leave and some substances to enter. In particular, the semi-permeable membrane allows water to both enter and leave the cell, but large molecules, like salt or sugar, are too big to pass through. The question is, how can we examine the semi-permeable qualities of this inner membrane with the hard eggshell in the way? Dissolve the outer shell by placing an egg in vinegar. Vinegar has a chemical called acetic acid. Eggshells contain calcium carbonate. When calcium carbonate in the eggshell comes in contact with acetic acid, a chemical reaction occurs. Calcium carbonate will break down to form carbon dioxide, water, and calcium acetate, thus dissolving the calcium carbonate shell. Within seconds, you'll see bubbles form. These bubbles are carbon dioxide that are produced when acetic acid breaks down the eggshell. Within 3 days, you should have a semi-permeable membrane free of the hard outer shell to conduct your experiment. Before starting our experiment, we will first mass our egg using a scale. Egg A has a mass of 94 grams. Egg B has a mass of 103 grams. Egg A we will place in a hypotonic solution, water. Egg B we'll place in a hypertonic solution, thick and sugary corn syrup. Egg A now has a mass of 97 grams. Egg A increased slightly to 103% of its original weight. We can conclude when a semi-permeable cell is in a hypotonic solution, water will enter the cell. Egg B now has a mass of 59 grams. Egg B decreased to 57% of its original weight. We can definitely conclude that when a cell is in a hypertonic solution, water will leave the cell. In fact, this is evident by the layer of water sitting on top of the corn syrup. However, 103% change in mass is not very significant. So how about we try this experiment again, but switch the eggs? This time, egg A is going in the hypertonic solution, which is still on the right. Egg B is going in the hypotonic solution, which is still on the left. After several hours, mash your eggs again. Egg A went from 97 grams to 55 grams. Egg A has decreased to 57% of its original weight. Egg B went from 59 grams to a whopping 105 grams. Egg B has increased in weight to 177%. From this experiment, we can definitely conclude that when a cell is in a hypotonic solution, like water, water enters a cell. On the other hand, when a cell is in a hypertonic solution, like corn syrup, or more realistically salt water, water will leave the cell. Now that you're done with this experiment, time to play with those shellless eggs. No! You're just gonna...
Module 3 of Chapter 3, Cellular Transport, will be continued on a second video.